Good morning, students. This is G.RAMA JL from G.RAMA Society, and welcome to webinar 4th session. Today, we are going to discuss about the photorespiration. Before this photorespiration, we have completed light reaction, which cyclic and non-cyclic, and dark reactions are completed. And next to that, the important topic is photorespiration. We will discuss it in detail. Before that, we need to know why exactly and how this process takes place. In normal cases, CO2 is accepted by RUBP, and for this adding the enzyme, helping enzyme is Rubisco. And this enzyme that acts as carboxylase, and sometimes it also acts as oxygenase. And when it acts as a carboxylase, it helps in combining CO2 with RUBP. Next, that means for this photorespiration, Rubisco enzyme is important. That we are understanding in this. So this Rubisco behavior changes. That means I just only told you that Rubisco enzyme acts as carboxylase and oxygenase. And this changing behavior, Rubisco behavior changes according to the environmental conditions like oxygen and temperature. Based on these two factors, it tries to change its behavior. Then, when the conditions are, that means about this oxygen and temperature, when these conditions like a high oxygen concentration and a high temperature, under these conditions, Rubisco behaves as oxygenase. When the atmospheric oxygen concentration is high and the temperature also high, at that moment, this Rubisco behaves as oxygenase. And it would cause, that means under this influence of oxygenase nature of a Rubisco, it would cause the combining of RUBP with the oxygen. That means oxygen is taken in by RUBP. And in this process, that means acceptance of O2 by RUBP results in the process of some different compounds which will be synthesized by the end of the process. And by the end of this process, that means acceptance of oxygen by RUBP, some new compounds or some different compounds will be synthesized. Along with this, by the end of this process, carbon dioxide will be released. And these changes are quite opposite to the C3 cycle. In C3 cycle or Kelvin cycle, the changes are just opposite to this changes. That means CO2 is accepted by RUBP and O2 will be released during the Kelvin process or C3 process. But in this, oxygen is accepted by RUBP and CO2 comes out as the end product. So this is about the introduction for photorespiration. We recollect about this before studying this photorespiration, just recalling for this Kelvin cycle or C3 cycle. In C3 cycle, Rubisco acts as carboxylase. It means RUBP binds with CO2 and prepares glucose. This is the part of a photosynthesis. That means here, in photosynthesis, CO2 is taken in and O2 is given out. Through this cycle, we can understand this, but because already it was discussed, so you might have grip on this topic. So even though, once again, observe the cycle. CO2 is accepted by ribulose biphosphate, RUBP. And the first product is phosphoglyceric acid. And this, by undergoing this phosphoglyceric acid, formation comes under carboxylation. And before that, C3 cycle takes place in 3 steps. First one is carboxylation, second is reduction, and third is regeneration. In carboxylation step, carbon dioxide combines with RUBP and results in the formation of phosphoglyceric acid. And this phosphoglyceric acid, by undergoing various steps, results in the formation of aldehyde, that is glyceraldehyde phosphate. And this changes comes under the reduction stage or step. And after the formation of aldehyde, few molecules, that means 3/4 molecules goes in a way and 1/4 goes in another way. And this 1/4 molecules, they helps in the formation of a glucose. And 3/4 molecules, they helps in regeneration of RUBP by undergoing various changes by the influence of enzymes and the utilization of ATP molecules. Those aldehyde molecules helps in the conversion of RUBP, which helps again in continuing the cycle. So this Kelvin cycle, in this CO2 is accepted and O2 during the non-cyclic photophosphorylation, photolysis of water takes place, and as a result of photolysis of water, oxygen as a byproduct, it comes out or it diffuses out. So here in C3 cycle, very clearly we are understanding that CO2 molecule is absorbed by RUBP, or CO2 binds with the RUBP, and that means the CO2 comes takes in and oxygen comes out. This is the normal process in C3. And in this C3 cycle, and also in light phase, during light phase, ATP molecules and reducing power both are synthesized. In this also, ATP molecules and reducing power are synthesized. And next to that, about Rubisco enzyme, just look at this picture. And this, during the normal C3 cycle, just only discussed, CO2 is accepted by RUBP, and helping enzyme is Rubisco. And here, Rubisco acts as a carboxylase, or it also acts as oxidase enzyme. That means that during C3 cycle, enzyme acts as carboxylase because it combines with CO2 with RUBP, and the entire dark phase reactions takes place where glucose synthesis takes place. Okay, then next, but Rubisco changes its nature according to the environmental factors like oxygen and temperature. And when the conditions like a high oxygen concentration and high temperature, Rubisco behaves as oxygen is. These are also just only discussed. And when the Rubisco acts as oxygenase, under these conditions, RUBP, instead of binding with the CO2, it binds with oxygen. That means uh, O2 is taken in by RUBP and CO2 is given out. So these are the important characteristic features of Rubisco, and that was observed in this also, in this picture or in this cycle. See here, this is Calvin cycle and this is photorespiration. In Calvin cycle, RUBP accepts the CO2 in the formation of a phosphoglyceric acid, and by undergoing a reduction, this PGA turns into phosphoglyceraldehyde molecules, and this aldehyde molecules again helps in the regeneration of RUBP and giving starch molecules. So here oxygen comes out in this. Oxygen molecule comes out as a byproduct, and CO2 definitely binds with RUBP. This is a normal process in Calvin cycle in the presence of this Rubisco enzyme. Whereas in photorespiration, RUBP combines with oxygen in the presence of RUBP oxidase. Whereas in Kelvin cycle, this carbon dioxide binds with RUBP in the presence of Rubisco or RUBP carboxylase enzyme. Why? Because CO2 binds with RUBP. For this, carboxylase is needed. Whereas in photorespiration, instead of carbon dioxide, oxygen is combining with the RUBP in the presence of, for this enzyme is RUBP oxidase. And this oxidase helps in the formation of PGA, releasing CO2 molecule. And again, it helps in regeneration of RUBP. So here, oxygen is taking in and CO2 is coming out. That's why this RUBP acts as oxidase under the special conditions like that, like a high oxygen concentration and a high temperature. It acts as oxygenase. Yes. In normal respiration, that means it might be in plants and animals. Usually, respiration means taking of oxygen in and sending the carbon dioxide out. It is defined as respiration. But in photorespiration, what is happening? Here, oxygen is, in photorespiration, taking of oxygen in and giving out CO2. CO2 is coming out. Whereas in normal respiration, that is in plants and animals, so O2 is taken in and the CO2 is given out. But in photorespiration, taking of oxygen in and giving out CO2. It means both are looking same. But in presence of light, this photorespiration takes place. In this respiration and photorespiration, the same sequence is there, but this photorespiration that takes place in the presence of light. That's why it is called as photorespiration. And taking of O2 and giving CO2 is a problem for the plants. And in CO2, taking up oxygen and giving out of CO2 is practically it is a problem for the plants. And in this, the CO2 carbon comes from a carbon compound which has synthesized as a result of photosynthesis. So, what done by photosynthesis is undone by photorespiration. That means in photosynthesis, CO2 binds, CO2 is fixed, and glucose will be synthesized and oxygen comes out. Whereas in photorespiration, oxygen is coming in and CO2 is coming out. So that means whatever CO2 is fixed in the during the photosynthesis will be utilized in photorespiration. That's why it is stated. So whatever is done by photosynthesis is undone by photorespiration. That means CO2 is fixed in photosynthesis, whereas it is utilized, whereas it is released in photorespiration. And this type of nature will be observed mainly in temperate region plants like wheat, rice, and barley. So in temperate region plants, we can observe this photorespiration very clearly. And the main examples are wheat, rice, and barley. Then coming into the photorespiration. So we can say that all those plants which performs normal C3 cycle, whenever they faces a high temperature and high concentration of oxygen, under these conditions, Rubisco acts as Rubisco oxygenase or RUBP oxygenase. And due to this, whatever CO2 is fixed by photosynthesis will be released by photorespiration. That means whatever CO2 is fixed by photosynthesis will be released by photorespiration. So this is a problem for the plants which lives in temperate regions. And now coming to the process of photorespiration, how it leads. See, this process actually it completes in 3 different organelles. That means it involves, this photorespiration process involves 3 organelles, like one is chloroplast, second one is peroxisomes, and third is mitochondria. And let us observe the changes in chloroplast. In chloroplast, C3 cycle takes place, which is a normal process, and in which Rubisco accepts CO2, and here it plays as Rubisco carboxylase or RUBP carboxylase. That means the first in prior slides we observed the C3 cycle in which RUBP acts as carboxylase. Why? Because in C3 cycle CO2 is accepted. CO2 is accepted by RUBP. That's why these changes are controlled by enzyme RUBP carboxylase. The next, this the first produced first product is phosphoglyceric acid, and this phosphoglyceric acid that furtherly changes or that furtherly undergoes various changes, chemical reactions, and results in the formation of phosphoglyceraldehyde phosphate. So this PGA turns into PGAL, and 3/4 particles are diverted in regeneration of RUBP, and 1/4 are useful or utilized in the synthesis of glucose. So here, this is a normal process of Kelvin cycle or C3 cycle. That means it is started with the binding of CO2 with RUBP in the presence of RUBP carboxylase, and the first formed product is phosphoglyceric acid, which is a 3C compound. That's why it is called as C3 cycle. And this PGA, that by undergoing various changes, it finally produces aldehyde. And in this aldehyde molecules, 3/4 molecules are utilized in the regeneration of RUBP, and 1/4 helps in the synthesis of glucose. This is a common procedure in C3 cycle. But under extreme conditions or under some special conditions like high temperature and high O2 concentration, this RUBP binds O2, under this high temperature and high oxygen concentration. RUBP binds with oxygen, where RUBP acts as RUBP oxygenase. So, under these conditions, Rubisco acts as oxygenase, and which helps in binding oxygen with RUBP. As a result of this compound, in presence of RUBP oxygenase, results in the formation of a compound, results in the synthesis of a compound which is called as phosphoglycolate. Next, that means in all these changes are taking place in chloroplast. That is important. Then next, this phosphoglycolate changes into glycolate, and this glycolate diffuses into peroxisomes. There is a next organelle where, see here, peroxisomes is a next. Peroxisomes is a next organelle of this process. So in chloroplast, once again, try to remember, in chloroplast, under extreme conditions like a high temperature and a high oxygen concentration, RUBP binds with the oxygen in the presence of RUBP oxygenase and results in the synthesis of phosphoglycolate. And this phosphoglycolate turns into glycolate, and this glycolate diffuses into peroxisomes. Now, let us see the changes in peroxisomes. Here, two more points in chloroplast. This change of a phosphoglycolate into glycolate, and this diffusion of a glycolate into peroxisomes, it's okay. Before that, because of the formation of this glycolate, this cycle is called as Glycolate Pathway. Just students, observe this. In chloroplast, glycolate was synthesized from phosphoglycolate, and because of this synthesis of glycolate, this pathway is also called as Glycolate Pathway. And this glycolate is a 2C compound. It is a 2-carbon compound. That's why it is also called as C2 cycle. Just remember the C3 cycle. Why it is called as C3 cycle? When RUBP combines, or RUBP binds CO2, and in the presence of RUBP carboxylase results in the formation of phosphoglyceric acid, which is a 3C compound. That's why that first formed compound was 3C compound. That's why they named the cycle as C3 cycle. Originally, it was discovered by Melvin Kelvin. So C3 cycle or Kelvin cycle, both are same. Here, in this photorespiration, which was taking place in 3 different organelles, in which the first organelle is chloroplast. In chloroplast, some changes are taking place in which phosphoglycolate changes into glycolate, and because of the formation of glycolate, this cycle is treated as glycolate pathway, or the cycle is also called as glycolate pathway. And this glycolate is originally a 2C compound. That's why it is called as C2 cycle. Now this glycolate diffuses into peroxisomes. Let us see the changes in peroxisomes. In peroxisomes, glycolate changes into glyoxylate, and by adding one amino group to this glyoxylate, it changes into glycine. And this glycine diffuses into mitochondria. So changes in peroxisomes are changing of glycolate into glyoxylate, and adding of amino group to this glyoxylate changes into glycine. So adding of amino group to the glyoxylate results in the formation of a glycine. Next, this glycine diffuses into mitochondria. So here the changes are simple. Changing of glycolate into glyoxylate and adding of amino group to this glyoxylate results in the formation of glycine. And now this glycine diffuses into mitochondria are the important changes in peroxisomes. Let us see the changes in mitochondria. In mitochondria, there's 2 molecules of glycine, because this is a glycolate is a 2C compound, and glyoxylate and glycine are also 2 carbon compounds. So 2 molecules of glycine changes into, or it splits into 2 different molecules. Those are 1 molecule of serine and another 1 carbon molecule, which was separated from glycine, comes out in the form of CO2. That comes out in the form of CO2. That means O2 is taken in photorespiration, and CO2 will be sent out. This was proved. And during this change, that means glycine into serine, amino group, that means conversion of glycine or changing of glycine into serine. Again, this amino group is separated, and that again diffuses into peroxisomes. Initially, amino group which was in peroxisomes that combined with the glyoxylate in forming glycine. And after diffusion into mitochondria, this glycine again it splits into this serine furtherly that undergoes changes, but the second molecule of carbon that comes out in the form of carbon dioxide. And here during this change, glycine releases, or the changing of glycine into serine, amino group will be separated, and again it diffuses into the same place, peroxisomes. And that amino group again it is ready to help in the synthesis of glycine. That means again it is ready to combine with the glyoxylate, and in the synthesis of glycine. So this is a change in mitochondria. First of all, in mitochondria, glycine two molecules that splits into two different molecules. One molecule is serine, another molecule, that means another carbon, C molecule, that means carbon molecule, that comes out in the form of carbon dioxide. So here once we should remember that oxygen was taken in and CO2 will be sent out. During this conversion, or during the change of glycine into serine, whatever amino group was added to glycolate is separated from the glycine and diffuses into peroxisome, and that helps in the continuation, or that helps in circulation of the cycle. So peroxisomes and that amino group help in the synthesis of again glycine. That means amino group is circulated between peroxisomes and the mitochondria in regenerating this glycine again and again, or in continuing the cycle. So these are the changes in mitochondria. So important steps in mitochondria once again. Glycine, which was formed or synthesized in peroxisomes, diffuses into mitochondria, and this glycine is a toxic compound, and it splits into, or it will be separated into two different molecules. One molecule is serine, the another carbon molecule that comes out in the form of carbon dioxide. By this, we are able to say it that oxygen was taken in and CO2 is going outside. And during this process in mitochondria, glycine, changing of glycine into serine, this glycine separates amino group, and this amino group again enters into peroxisome. And this is focusing what this amino group is again ready to combine with the glycolate to form the glycine. That means this amino group is helping in helping in regenerating glycine, and also this amino group is circulated between peroxisomes and the mitochondria. These are the changes in mitochondria. See here, schematic representation of photorespiration. Here, oxygen combines with RUBP. In the presence of oxygen is RUBP oxygen is, and the resultant is PGA, and this acid will be converted into aldehydes, and regeneration of RUBP and release of carbon dioxide. So here the first formed thing is C2 compound, R2C compound. That's why it is called as C2 cycle. So this is about the schematic representation. The next, here the changes are exhibited in separate cells. In chloroplast, oxygen is combining with the RUBP, resulting in the formation of phosphoglycolate or PGA, and this PGA turns into glycolate, and this glycolate diffuses into peroxisomes, and glycolate by adding with the amino group turns into glycine, and glycine diffuses into mitochondria. And in mitochondria, this amino group is separated in the form during the formation of serine molecule 1. Another carbon molecule is released in the form of carbon dioxide. So here CO2 is released, O2 is taken in. Then next, this serine, whatever is formed, is diffused into peroxisomes. And 1 minute. Yes. Serine diffuses into peroxisomes. In peroxisomes, it changes into pyruvate. That means serine changes into pyruvate, and pyruvate changes into glycerate, and this glycerate enters into chloroplast. By utilizing one ATP molecule, it is converted into PGA. And it is considered as this photorespiration is considered as a harmful process. Before discussing that, see the changes in picture. See here, serine, whatever is synthesized, enters into peroxisomes, and this serine is converted into pyruvate, and this pyruvate changes into glycerate, and this glycerate diffuses into again chloroplast, and by the utilization of one ATP, this glycerate turns into PGA, and this PGA again helps in the regeneration of RUBP, and this RUBP again combines with oxygen and resulting PGA, which is a 3C compound. Again, the cycle will be started. So here the changes are once again oxygen, that means under extreme conditions like high temperature and high oxygen concentration. Oxygen is accepted by RUBP in the presence of RUBP oxidase enzyme, and this results in the formation of 3C compound, phosphoglyceric acid, and this changes into glycolate, and this glycolate enters into peroxisomes, and that is changed into glycolate, and when it combines with amino group changes into glycine, and glycine diffuses into mitochondria, and in mitochondria this glycine two molecules are changed into two different molecules by relieving amino group, and this relieved amino group again diffuses into peroxisomes, and the separated two molecules out of two, one molecule is serine, another carbon molecule comes out in the form of carbon dioxide. Now this serine from mitochondria diffuses into peroxisomes, and in this peroxisomes, serine is changed into pyruvate. Pyruvate changes into glycerate, and this glycerate diffuses into chloroplast. In chloroplast, by utilizing one ATP, it turns into PGA, and which results in the formation of RUBP. Again, this is treated as regeneration of RUBP. So again, it starts the same procedure. So in this way, this photorespiration will be completed. And this photorespiration, it is treated as a harmful process. Why it is treated as a harmful process? Means in this, before this cycle, let us recall the C3 cycle. In C3 cycle, 18 ATP molecules are framed, are synthesized, and 12 NADPH2 molecules are synthesized. And in addition to that, that means in C3 cycle, energy count is 18 ATP and 12 NADPH. But whereas in this photorespiration, there is no synthesis of ATP. There is no synthesis of ATP. So, and no reducing power is synthesized. So here, energy molecules are not synthesized. That's why it is treated as a harmful process. That means no formation of ATP and no formation of reducing power. That's why it is treated as a harmful process. This process is seen in temperate region plants, predominantly in C3 plants. And there are some other plants which shows some adaptation. So to avoid this photorespiration are called as C4 plants. That will be discussed in further chapters. But before that, this photorespiration is not helpful. It is harmful to the harmful process. It is treated as a harmful process. Why? Because no energy forms are synthesized. That's why. Whereas in C3 cycle, ATP and NADPH molecules are synthesized, will be synthesized. That's why it is useful. And so in this way, photorespiration when the C3 cycle is not possible, whenever the conditions are extraordinary, that means with a high temperature and a high concentration. Under those conditions, Rubisco plays as RUBP oxidase, RUBP oxygenase. And due to this conditions, O2 will be absorbed by RUBP and the CO2 will be released. So if we observe the process of photorespiration, it takes place in 3 different organelles. First one is chloroplast, in which Rubisco acts as a oxygenase. Under these conditions, RUBP accepts with oxygen and results in the formation of phosphoglycolate, and this phosphoglycolate changes into glycolate, and glycolate diffuses into peroxisomes. And because of this glycolate synthesis, and this cycle is also called as glycolate pathway, and this glycolate is a 2C compound. That is important. This glycolate is a 2C compound. That's why it is called C2 cycle also. And next, this glycolate that diffuses into peroxisome. So with this, chloroplast changes will be stopped. The next, in peroxisomes, if we observe the changes, this glycolate that changes into glyoxylate, and glyoxylate changes into glycine. And what way means? By adding the amino group to the glyoxylate, it is converted into glycine, and this glycine diffuses into the mitochondria. And this glycine molecules are 2. Number of glycine molecules are 2, and they, out of these 2 molecules, they splits into 2 different molecules. One molecule will be converted into serine, and another molecule of carbon that comes out in the form of carbon dioxide. By this, we can say during this photorespiration, oxygen is taken in and CO2 will be sent out. That's why this photorespiration is treated as a harmful process where energy currency is not synthesizing. Whereas in C3 cycle, 18 ATP molecules and 12 NADP molecules are synthesized. So here, during this changing of glycine into serine in mitochondria, this glycine releases amino group into the mitochondria only. But this amino group diffuses after relieving from glycine, it moves into the peroxisomes. And that is, amino group will be ready to accept or ready to combine, ready to bind with again a glycolate, glyoxylate to form the glycine. Okay, next, in peroxisomes, the serine, whatever serine is synthesized in mitochondria diffuses into peroxisomes. And in this peroxisomes, this peroxisomes. This serine changes into pyruvate, pyruvate changes into glycerate, and that glycerate diffuses into chloroplast. Again, this chloroplast, by utilizing one ATP molecule, converted into PGA, and this PGA again helps in the formation of RUBP regeneration, and this RUBP is ready to accept oxygen molecules. In the presence of RUBP, oxygen is to form again the same products, that means a phosphoglycolate. So in this way, the cycle will be repeated, and this process is seen only, this photorespiration is observed only in temperate region plants, and there are some more plants which shows some adaptations to avoid this photorespiration, and those plants are called as C4 plants. And about C4 plants discussed in further classes. So this is about the photorespiration. Thank you.